I would like just now to invite Richard Kam to come up. Richard obtained his PhD from the Chinese University of Hong Kong in 2011, and after his postdoc training, he joined the Hong Kong Baptist University Hospital in 2013 and was a staff in charge of the proteomics laboratory for systems biological research. Richard's research interests include clinical applications of quantitative proteomics, steroid hormone analysis, and pharmaceutical bioanalysis using LCMS techniques. Richard.
Thanks for the kind introduction. So I'm happy here today to share with you some of our recent development, which is the top-down analysis of transferrin glycoform using the driver spot. So let's see if it puts. Yeah. So I won't be covering the physiological role of transferrin in circulation because it will take a very long time to cover. But I'll talk about the protein structure as well as the glycosylation about this particular protein. So transferrin is mainly synthesized from the liver. So this is a kind of a standard glycoprotein in circulation, but also it can be found in CSF because the brain also synthesizes a brain-type transferrin as well. So it's an iron-binding protein with a single polypeptide chain with two domains in duplicate in the protein sequence. So it has two well-documented N-glycosylation sites with a very well-defined N-glycan structures. And the intact transferrin with two glycosylation has an approximate molecular weight of about 79 kilodaltons. And of course, the glycosylation pattern is related to many pathological conditions. For example, the congenital disorder of glycosylation, the CDG. And also the glycosylation pattern is found to be affected in different kinds of the liver derangement, for example, in fibrosis or cirrhosis. And also, of course, there is a carbohydrate deficient transferrin in the event of the chronic alcoholism. Okay, so this slide is very important because it is the language we will be using in this presentation. So the N-glycosylation is a structurally defined post-translational modification. It's determined by the protein sequence itself, the ER function, as well as the Golgi apparatus. So about the DNA and RNA as well as the protein are synthesized based on a template. And then the N-glycosylation is synthesized based on a combination of all these factors, as well as the presence of all different kinds of the glycosyltransferase. But regardless of how they are synthesized, the glycosylation always carry a common core structure. The common core structure is shown here. It's 2 units of the N-acetylglucosamine or N-GLUNAC combined with 3 units of the mannose residue, the green circle here. And the N-glycosylation can be broadly classified into 3 types. The first type is the high mannose, which is mainly comprised of all the mannose residue. And another type is the complex type. The complex type is comprised of N-acetylglucosamine, followed by a galactose, which is a yellow circle, and then with some of the event, they carry a sialic acid, which is represented by the purple rhombus here. And the last type is the hybrid type, which is a combination of the high mannose and the complex type antenna on the N-glycosylation. And apart from all these 3 types, it can be modified by a fucoxylation. This is the attachment of a fucose on the first unit of the N-acetylglucosamine, which is, this is here. And also it can be modified by an addition of a bisecting N-acetylglucosamine in this position. So this is the main type of the N-glycosylation found in different kinds of the glycoproteins. So of course, the glycosylation structure of the transferrin has been studied in many different cases, for example, in CDG. And in fact, the people are only really interested in the glycosylation pattern, but not the transferrin protein itself. So the transferrin is mainly can be taken as a carrier of the glycosylation and the defects associated with different pathological conditions. So other glycoproteins are also studied in the CDG, for example, the apolipoprotein C3, because it only carries the O glycosylation, which is kind of useful when we try to differentiate between CDG or the defects in the liver derangement. And also there are also some studies on the transferrin variant because it may compromise the interpretation of the glycosylation pattern. And quite interesting, in 2004, there is a paper using a technology of the DNA sequencer. So they studied the total serum glycomics for the differentiation of the liver cirrhosis. So what they did is that they removed all the N-glycosylation from all the serum glycoproteins and then compare the pattern of this glycomic signature in normal subject as well as those with liver cirrhosis. And they found that it's quite a useful marker as a non-invasive diagnosis of liver cirrhosis, which is very interesting. Okay, so here's the structure of the human transferrin, the schematic. So the first 19 amino acid is the signal peptide, followed up by about 670 something amino acid. So they can be divided into two domains. The domain 1 is the N loop, while the domain 2 is the C loop. And the two glycosylations are attached to the arginine, number 432, as well as, the asparagine, the number 432, as well as the 630. So these are the two N-glycosylations. So the native form of the transferrin in circulation is the tetracellulose, which is the native form. And we also found some of the pentacellulose in normal subject. And these two kinds of the transferrin can both be fucoxylated with an addition of the fucose residue. And we can also find some of the tricellulose transferrin, as well as a very small amount of the dicellulose transferrin. And some of them, of course, can also be modified by a bisecting N-guanide, as well as we can also find some of the decellular transferrin in the case of the CDG. So people have been using the isoelectric focusing to study different transferrin glycoforms for a long time. So by using the IEF, they can separate the different transferrin glycoforms based on the number of negative charge. And that actually corresponds to the number of sialic acid attached to the glycoforms. So for example, in a control case, we will see a very dominant band of the tetracellulose transferrin right here. And then for the CDG type 1 patient, the dicellulose transferrin as well as the acellulose transferrin will be elevated. And the tetracellulose form will be kind of decreased in this case. So this is actually a very early example of the use of a clinical glycoproteomics to study the different transferrin glycoforms pattern. And then in 2001, there is a paper describing the use of an automated LCMS platform with an online immunocapture technology. So the serum sample can be captured online by using an immunoaffinity column and then eluted into the LCMS instrument for analysis. So transferrin is a very large protein. So in the MS spectrum, it will become a multiple charge species. So this is what we found in the mass spectrum. So all of these are the multiple charge species. And then they use an algorithm of the deconvolution to generate the intact mass of the transferrin, which is about 69 kilodalton. And this corresponds to a tetracellulose transferrin, as shown here. And then by using a deconvolution algorithm, they can also identify the monoglycosylated transferrin in the case of type 1 CDG, which is shown here. And also the deglycosylated transferrin can also be identified by using this method. So what we did is basically very similar to what is described in this paper. So our method can be used for serum or CSF. And for the dropper spot, we found that it's actually quite easy to do the dropper spot. So we only need 2 punches of a 3.5 mm diameter punch, and then extract it using the phosphate buffer saline. And the eluate should be sufficient for the affinity capture. So we use an offline immunoaffinity capture. The material we use is from Thermo Fisher, the Capture Select, which is quite good product. The binding and elution is quite easy. And then the transferrin is subject to LCMS analysis by using a high resolution time of flight. And we also use a deconvolution algorithm by Waters. It's called the MaxSense1. And we will then get the exact mass of the protein species. And then we'll screen for the accurate mass screening and predict the glycoforms in the transferrin eluate. And we also can get a relatively semi-quantitative of the different glycoforms in the circulation. So the algorithm we use is the MaxSense1 or maximum entropy. Now the mathematical principle is I don't fully understand, to be honest. And it's a kind of a black box from Reuters. So is there any Reuters representative? Maybe they can enlighten us. So, but in short terms, it aims to find the most simplistic probability distribution. That is to maximize the entropy, which is consistent with the known input information. So for this sentence, I don't really understand what they're talking about. But anyway, the raw data input, there's a multiple charge spectrum, is the question we provided to this algorithm. And then it will calculate the answer, which is the zero charge data. Okay, so here's one of the examples. So on the left-hand side is the intact transferrin mass spectrum we obtained from the top instrument. So of course, it's carry a lot of the positive charge, up to maybe 45 or 46. So it's a kind of follow a normal distribution right here. And if we zoom into one of the charge state, actually we can see a lot of the fine details, right? So it's not a single peak, but there's a lot of the minor peaks around the most abundant one. So this is a driver spot sample, and this is what we got after the deconvolution. So of course we can see the most abundant tetracellulose-transferrin with molecular weight of about 79 kilodalton. And on the bottom panel is the zoom-in region. And again, we can see a lot of the minor details, right? A lot of the minor peaks around here. And this is one of the very interesting things about glycoproteomics is that we can calculate the mass difference and then predict the glycostructures. So this is the most abundant one, the 79 kilodalton. And then this minor peak is exactly 2,206 dalton. And then this kind of peak is, again, another 2,000 something dalton. And this mass actually corresponds to one intact complex type of the N-glycosylation. And then we also see a minor peaks with a difference of a sialic acid. And then this peak over here has an extra antenna with a 656 dalton. And then this peak next to this one has a mass difference of a fucose. So based on this information, we can actually predict the glycosylation structure of all of these minor peaks. So at the end, what we have here is something like this, the tetracellulose form, the most abundant one. This minor peak is the tricellulose form. And then this peak is the monoglycosylated transferrin, lacking one of the complex type N-glycan. And then this peak, the 80,000 dalton peak, is the pentacellulose transferrin with a fucosylated species right next to it. And actually, we can also see a very minor peaks. This is the fucosylated tetracellulose transferrin right here. So of course, this is just a prediction based on the mass screening. We can also do some enzymatic digestion to get some insight into the structures. So the PNGSF is the very useful enzyme. It's very specific to remove N-glycosylation from the protein. And we use a condition that is called a non-denaturing condition, which is for transferrin, one of the glycosylation sites is actually resistant to PNGSF digestion. And then by using a non-denaturing condition, we can remove only a single glycosylation structure from the transferrin. Neuraminidase can remove the sialic acid completely, and then also we can do a combined digestion to see what will happen. So after PNGSF digestion, we can generate a monoglycosylated transferrin right here. So this mass also, our predicted structure is also a monoglycosylated transferrin. As for the neuraminidase, we can remove all of the sialic acid and give us this peak. And then the combined digestion will give us a monoglycosylated acellulose transferrin. And the glycan structure also is consistent with our enzymatic digestion. And it's also the same for the pentacellulose form. So the pentacellulose transferrin is right here. So this minor peak is the monoglycosylated tricellulose transferrin. This peak is the this one. This one is the completely decellular transferrin. And then finally, in the combined digestion, we'll obtain a monoglycosylated triantennary transferrin. So in this way, we can also study different types of the glycosylation structure in case we need to confirm the structure of this transferrin. Okay, so we've compared the glycoforms obtained from the driver spot as well as the pair serum. So the pattern is more or less quite similar. And again, we can identify most of the dominant species, including the tricialoform and the tetrasialoform. And if we compare the relative ratio between the pentasialoform and the fucoxylated species, we can see that it is more or less quite similar in the driver spot as well as in the serum samples. Okay, so this is another case of the driver spot and the paired serum. This case is a little bit different because this is a patient carrying a transferrin variant. So the molecular weight is about 30 Dalton larger than the wild type. And this patient is a heterozygous variant, so we'll see a double peak for the transferrin. Right. So there are two kinds of the polymorphism that can give rise to this variant. And without genetics, we don't quite, we can't confirm whether which one is the polymorphism we are looking at. But still, we are able to visualize the variant readily in the LCMS. And again, the pentasialoform as well as the fucoxylated species, the ratio is more or less similar. So from this point on, we can see that the driver spot and the serum are more or less has the same behavior in terms of the transferrin glycoanalysis. So the variant has been quite a problem for the isoelectric focusing interpretation. So for example, in this case, the control IEF pattern is something like this. But then in this patient, he has a homozygous variant. So we see that the band actually shift a little bit downward. And without this information on the presence of the variant, we may see that there is a presence of a tricialotransferrin in this IEF pattern. And then on the right-hand side is the patient with a heterozygous variant. And again, there is a double peak here, a double band here. It may suggest that there are presence of a tricialotransferrin. But in fact, this patient is actually carrying two types of transferrin, and it can be only visualized by using a neuraminidase treatment on the IEF, before the IEF. So for IEF to differentiate between the true case of the CTG as well as the variant patient, the neuraminidase treatment may be necessary. But for the LCMS, we can already readily visualize the presence of the variant and avoid this kind of a confusion in interpretation. So we have studied the relative abundance of different transferrin glycoforms on the driver spot as well as in the serum samples. Now the correlation is not perfect, but at least it's more or less linear. So the linear relationship has been observed for different kind of the transferrin glycoforms, from dicialotransferrin up to a pentasialofucoxylated species on the low end of the linear curve. And for the tetrasialotransferrin, the relationship is also more or less linear. And for this correlation, we have also included some patient with a defect in glycoxylation so as to give a higher relative abundance of this dicialo and tricialotransferrin. So we have also investigated the linear relationship between different glycoforms. So we have made up our own transferrin standard, the dicialotransferrin standard, by using a PNGSF and then mix them together in a different ratio with a transferrin standard. And then we compare the observed percentage of the dicialotransferrin compared to a nominal percentage. And we found that the correlation is more or less quite linear. But we also observed that in order to have a linear input, a linear relationship, we will need a certain input of the transferrin for this analysis. So for example, we can see that the relationship for 20 microgram or 10 microgram transferrin input is more or less the same, which is the orange and the blue line. But then if we reduce the transferrin input to, let's say, 5 microgram, the linear relationship does not hold actually. And for 5 microgram input, you can see that the response is not really correlated with the observed relative abundance. And you can also see that the imprecision of the assay is gradually increased. So this method kind of required a certain amount of transferrin input to be valid. But nevertheless, at a very low relative abundance of a dicialotransferrin, we can still see a very discernible peaks at about 5.3% of a dicialotransferrin. We can still have a very nice signal-to-noise ratio right here. So we can still identify the dicialoform, but not for quantitative purpose. Okay, so because we don't have many positive cases in the driver spot, and there's also no QC material yet, so we have to rely on the EQA scheme to assess our performance. So on the right-hand side is the two material from ERDIM EQA. So the first case is a mixed type 1 and type 2 CTG pattern. And again, we can see the dicyanotransferrin right here, as well as the presence of a very low amount of the decarboxylated transferrin. And also there is a very minor peaks here. And based on the accurate mass screening, we can see that this is a biantennary truncated form of a monoglycosylated transferrin. And then on the bottom one is a type 1 CTG with protein polymorphism. And again, the dicyanotransferrin, we can identify them quite reliably, as well as the presence of a double peak, which confirms that there is a protein variant in these samples. And then on my left-hand side is there are two positive cases in our hospital. So the first one is a 3-year-old boy with global developmental delay. So this is a serum sample. And again, we can see the presence of a very strong peak of a dicyanotransferrin, as well as a minor peak of the decarboxylated transferrin, which suggests that the patient may have a type 1 CTG. And then on the bottom panel is quite interesting. It's actually a nasal discharge from a 57-year-old gentleman. So the clinician want to confirm whether this patient has a CSF leakage. And from the glycoform analysis, we can see that there are actually a presence of these two types of the acyanotransferrin with different glycosylation structures. So actually, there are brain-type transferrin is only found in the CSF. So we can confirm that the nasal discharge of this patient is actually CSF leakage. And this spectrum is actually generated from a raw data, something like this. So you can see the signal to noise is not really good, and there's quite a high level of background right there. But still, we can generate quite a decent deconvoluted spectrum for interpretation. Okay, so we have also done a proof of concept study just to see if our assay can be used for the investigation of chronic liver disease and alcoholism. So we are not trying to do a biomarker study. It's not really a sophisticated study, but it's just a very preliminary study to test our assay. So we have found some of the patients by retrospective analysis. We select some of the patients with chronic liver disease based on the patient history, the ultrasound finding, like the ultrasound scan or fibroscan. And also we have checked the biochemistry for elevated liver function for more than 2 months. And we aim to find if there is any observable difference in the monoglycosylated species as well as the fucosylated species. It's because fucosylation has been known to be increased in the case of the liver function derangement and liver fibrosis. So we want to see if we can also see this kind of difference in our cohort. So we calculate the monoglycosylation ratio, which is the response of a monoglycosylated transferrin, which is this one, compared to that of a response of a diglycosylated transferrin. And the fucosylation ratio will be the response of all the glycosylated species, which is those with a red square, for example, this one and also this one, compared to all the response of all the other species. So we do see a difference between normal subject and those with chronic liver disease and alcoholism. Now the difference is not really that great because it's not a very reliable biomarker, but still we can see a difference. And the difference between the fucosylation ratio as well as the monoglycosylation ratio as well. And from the mass spectrum, the top panel is those from a normal subjects. So we can see that the fucosylated species is not present. And then those with chronic liver disease, the fucosylated species are elevated. And for the monoglycosylated species, also we can see a slight increase in the case of the chronic liver disease, although the increase is not really pronounced compared to normal subjects. Okay, so as a brief summary, we can detect the different kind of transferrin glycoform, including the neutral species and those with fucosylation in the dry blood spot by using a top-down approach. So this is very important because the IEF cannot differentiate between neutral species, and they are also counting the number of sialic acid, so they cannot really differentiate between decarboxylated transferrin and acyanotransferrin. And we also show that the dry blood spot is quite a viable alternative matrix because it has comparable behavior to that of the serum samples. And hopefully we can do some relative semi-quantitation of transferrin glycoforms and establish a reference interval on the relative abundance for the interpretation. So that's the end of my presentation, and I would like to take all the questions.
Thank you, Richard. You make us feel very inadequate. We measure CDT by HPLC UV. So, yes, we've got a long way to go. Just in the interest of time, we might have one quick question, and we've just got to keep moving on for lunch. Is there a quick question from the floor?
Thanks. Great talk. The place where your method would obviously really shine is in the CDG type 2 defects, which are the post-modification defects that are taking place in the Golgi, where you would actually be able to see, hopefully, identify where the particular defect was based upon the pattern you identify. Have you had any opportunity to look at any type 2 defects?
Unfortunately, no. We haven't seen a lot of the case of CDG type 2 because the CDG is kind of rare in Hong Kong. So we have to only rely on the EQA at this moment. Yeah.
Thank you.
But based on other literature report, I hope that we can see different kinds of glycoforms.
I might just like to invite our fifth speaker up, last speaker for this session. Sorry. Sorry, Rhonda. Thank you. Thank you, Rhonda. Thank you.